So as you are very well aware, there's many insect pests in this world, and in a publication that came out in 2017 from Kew Garden, at that time Helicoverpa armigera, so the cotton bollworm, was listed as the top pest, and this was listed based on the number of publications and research effort that's been done. Spodoptera fujiperta listed number 8. So I think if we are to review this list now, I think the fall armyworm will be number 1, given the amount of activities that's been done. But Bermesia tabesi, the whitefly species complex, is also a very challenging pest, and the challenging nature is due to their cryptic species identification, as well as their ability to transmit virus. So you guys all know about how to identify the fall armyworms. They do come in many different color morphs. In Brazil, where I first got introduced to this pest, they come in all sorts of colors, and the Brazilians are amazing at identifying. They can just look at it, they beat around the soybean crop, which are OGM, and they can find this is Fujiperta, this is something else, and all that. So they're very, very good at identifying. They deal with this pest forever. So a lot of the initial knowledge that I gained was through working with the Brazilians and helping them with the molecular aspects, and they taught me about the biological aspects and all that. So in the native range, millet is obviously what we are seeing as well. Cauliflower, they damage food crop. Guava, the maize, a lot of them are GM as well, and they do have resistant populations there. So that's a significant issues. And pasture grasses significantly damaged often. So if you can probably see in this picture here, you can see that at the far distance, those are the pasture grasses. Coming here is all stripped bare by the fall armyworms. It's just amazing. So what strains are they? They don't really care about what strains they are in Brazil. It's fall armyworm. They have to deal with it, and that's it. So in the invasive range, initial literature likes to tell us that there are different strains, the corn or the rice. We did a literature review commissioned by the Annual Review of Entomology, and I led the team, and after reviewing hundreds of literature, we decided that the idea about calling them a corn or rice is actually not helpful, because certain literature would say yes, they feed on corn, and certain literature says some population feed on rice, and therefore they're rice or corn. But that's because they read that and they're happy with what they have read, and this is what it is. But if you go through the literature, it's actually not that clear cut. So having a definition of it's corn or rice, it doesn't really help, especially in the invasive range. All right, and we'll demonstrate some, we'll provide some genomic evidence of why really trying to call them a rice or a corn or whatever strain that people like to call them in the invasive ranges just don't help with our ability to manage them. So interception, we know a lot about this pest being a global pest because in the EUFRESCO, so the European Phytosanitary website, you can download annual reports as well as monthly reports, and often you can see that they intercepted fall armyworms coming from North and South America, especially South America and Central America. In capsicum, for example, often it would be listed capsicum fall armyworms. Cut flowers is another big thing. In Australia, we look at the interception data, and fall armyworm was predominantly asparagus. So you like to buy your asparagus from your supermarket, from Peru, places like that. Often they come with fall armyworm. And tomato, they will attack tomato. So what strain is that? I don't know. And again, I don't think that it helps. To a certain extent, understanding the genomics and the signature of the population will help with our management, but to really force them into a corn or rice category is probably not helpful. And so we propose that we just call it C or R strain, and that's it. So a range of a range of crops in the invasive range as well. Some of them are obviously in Australia, sweet potato or rice. Actually, no, melons, capsicum, and so forth. But in China, there's a lot of literature in Mandarin, published in Mandarin. And they'll report barley being damaged, wheat. In Philippines, for example, and Vietnam, rice are being attacked. That's not an issue. We're moving away from rice anyway because of the water issue. But rice, and believe it or not, oil palm. So nursery in tropical countries, they're starting to see oil palm being damaged. So yeah, so you know, there people say, you know, so what strains attacking rice or oil palm or things like that? I don't know, I don't care, because they are hybrids. So a bit of the life stages you'll know, and I just talked to Ramesh, and what we see in the field, or what you guys see in the field, is literature. It's relevant. It's reflected quite accurately, the amount of time, the length of time it takes for per generation. But you know, like, lessons that we're learning from new places, like for example, when during COVID time we were having monthly meeting for the FAO Fall Armyworm Technical Group, we were trying to warn New Zealand, for example, that this pest is coming, this pest will be at your country, and they basically say in the literature they will die when the weather gets cold, we are not too concerned. So I just returned back from Christchurch. It was pelting down with hail, and very cold, 5 degrees. And they say if you go out to the field now, you see that they're quite happy there. So that's 5 degrees down south at Christchurch, which is the same latitude as Hobart. So they don't care. And we are actually working with Nepal collaborators. They said they're quite happy out there in 0 degree. So Australia took about 2-3 weeks to declare that this pest is uncontrollable. Anyway, and New Zealand is due sort of deciding whether they want to declare the pest, hoping against all hope, perhaps that the cold weather will kill them. But it's spread from North Island now down to South Island, so it's a fairly robust, quick, adaptable insect pest, and we have to reckon with that. There's a lot we don't know. So Australia being an island, we always thought they were quite insulated, and security-wise, we're quite safe. But movement, obviously, how they came to Australia, there's a big component with natural movements, obviously. There's no denying it. But how about movement back from Australia across, back up north into, say, Pacific Islands, into Papua New Guinea, Timor-Leste, Indonesia, and so forth? We actually do not know a lot. And Australia company has a lot of interest in those countries because they often have trial cropping experiments in those countries, so they have land that they use to grow the relevant crops. So how we actually manage this pest eventually will impact on other interests of Australia as well, and also across the global region. So CSR increasingly is focusing on how we can actually improve the ASEAN-Pacific, Indo-Pacific region biosecurity through cutting-edge science to help those regions, because eventually it doesn't matter how well we actually protect our border, how many hundreds of millions of dollars per year the federal government invests in it. If we don't help our neighbors, when they have a pest, eventually we pay for it. So that's what we are increasingly, CSR is using the science to link up with the Southeast Asian partner, with Indo-Pacific partners to better protect biosecurity across the Indo-Pacific regions, which will impact Australia as well. So going into understanding why there's such a pervasive, invasive, damaging pest. So a while back, so this must be 2016, 2017, we did the genome of the fall armyworm, as well as Helicoverpa armigera, Zaire, and trying to understand what makes them the pests that they are. So you can see that in this case, we actually do not know when they diverge from a common ancestor into Helicoverpa and Spodoptera. So we can't tell. There's very little, I guess, fossil evidence that we can go back. But what is obvious is that when you compare their genetic repertoire for detoxifying chemicals, for their ability to mutate and break down insecticide, so detoxifying genes in this category, you can see that in this noctuid, so the night moth category, they really have a lot of resources to really quickly adapt and eat just about anything. And when you compare that with, say, for example, Bombyx mori or other Heliconius and even Patella, so diamondback moth. So these are fairly polyphagous, and you can tell through their genomic signature. So is it a C or R, really? 2010 was when the TPI gene was advocated as a gene to use for understanding the C or R strain, and this was advocated by USDA. So they look at the TPI gene. The TPI gene is actually a sex chromosome gene. So in Lepidoptera, predominantly, males are the males and females are actually opposite to humans. So females are the heterogametic sex, so they only have one sex chromosome. So in this case, it's F, and the male have two copies, ZZ. And the TPI gene is actually located on the Z chromosome. So when you look at that, in this scenario that I'm showing, ZW, the female, has a COR. So let's just say that this is a hybrid female. Okay, it's an offspring of, if you like, a C and an R mating together. And their offspring between this mating pair, so both of them are hybrid, gives you, in this case, either ZZ, which is a CC. If you cross Z and Z together, you get the CC, or you get a CR. Okay? And if you have females, you have ZW and ZW, and it's because it's heterogametic sex, is only a single chromosome, you can only see a C or a R. So the red color there tells you that from hybrid individual using this marker, you get only a quarter evidence of hybrids. The other, when you sequence them, when you characterize them, they appear to be a purebred. So how can you have two hybrid mating together to give you a pure individual? So this marker is really, really problematic, and yet everyone is pushing for it. And we try to tell them in this annual review paper, and people are still using it. I get countless papers every month. Please review. And if I see that, I just say, I'm not going to review it. Go read the paper. And people just don't understand. They just keep pushing for that. So there are, ultimately, when we say it doesn't matter, but in the native range, it does matter a bit because they do have pure, when you look at the genome, pure C or pure R overall. It's fairly unmixed. And there are different mating activities. So mating activity is the first evolutionary barrier for speciation. So the C females, typically they are quite mating sexually active early at night, and the R strain females are typically active late at night. And when you cross a C female with an R male, typically they're less fecund and the offspring are not so fit as when you cross the other way around. So there are some differences. How that actually impact on the fall armyworm across its native range and invasive range, we actually don't know because we can't tell what actually came over. Is it hybrid that already came over, or did they come over and then they mate and they produce hybrids? So these are past history, and we really can't tell what actually happened before. But this paper is really downloadable. It's open access, so if you're interested to read more, please feel free. And it tells you what are some of the differences, sex pheromones, components, minor differences, whereas in the native range, there's actually more complex. And that is quite important because currently there's pheromones management options, strategies that people are trying to use. And places like, I think it was Nepal, Bhutan, I can't remember, they say that when they bought the pheromone from one country, it didn't work, and if they bought it from another country, it worked. So the question is, in your populations, do you have different different populations with different genetic components or not? Or is it because the lure truly don't work for one and then work for the other? So managing this pest is still at an early stage, and we do have a lot of hurdles and difficulties to get over. So these are all outlined in the paper, so please, if you're interested, have a read. And then, so I'm saying that TPI genes, everyone's trying to use TPI genes to understand whether it's a C or R. That's probably not the best gene. As you can see, you have a 25% chance of getting it right anyway, and your error rate is actually 75%, which is really, really poor. The other way to work out what they are is using the CO1 gene. So that's the gene that is typically called the DNA barcoding gene. And of this gene, about 15,000 base pair, 15,500 base pair typically for armyworms. We're only using about, I don't know, 6%, 600 base pair, 500 to 600 base pair. So what about all these other genes? We should be using them if we really want to understand what we're dealing with. And yet people are just using the barcoding region, which is a tiny front end, 600 base pair. And this region, this is a barcoding gene. So it's been developed for species identification. Beyond that, you're pushing the limit of your marker because it's such a short region of the DNA. So the other thing is actually this species, this pest, because it is a DNA barcoding species identification gene, so it's actually lacking variation. So when you analyze that without realizing that it's actually a region just for species, rapid species identification, you're using to infer population movements and whatnot, the lack of variation in some of the individuals that you're looking at give you the conclusion that because mitochondrial DNA, mitochondrial DNA is maternally inherited, so mother to offspring, so always females to offspring. So there's no recombination, and because they're all the same, you say that they're coming from a single mother. All right, so that's what happened when, in this case, there's just an example of a Chinese study very early on when they reported for armyworms. They again just look at the CO1 gene a little bit, and with that they say that it's identical. So it is identical to the one from Africa. It came from Africa, and that is quite a dangerous assumption because by the general community that kept saying that this is a pest that's coming from Africa, anything Africa has studied and reported, it will be the same for us. We are actually ignoring that the possibility that the pest is actually coming in through our own country and new populations coming in. And that's what we want to understand and have a better biosecurity to actually understand, do we have other leakage? And how, if we have other leakage, what happened? So potential distribution. So there are many studies trying to look at how populations are studied. CSIRO through Darren Kritikos developed the Climax. And so far the Climax, by comparison to the others, not because I'm from CSIRO, but truly when you look at the other methods of simulating where the pest might be, this seems to stand out. And this simulation, this program uses irrigation, global irrigation data and rainfall data as well, because water is really important for this pest. So in Australia, we can see that these are going to be persistent population up the north. So it's well within your region. So unfortunately, you're going to be facing with that, like it or not. And periodic seasonal populations might be experienced even down to Tasmania, and we can see that in pockets of New Zealand, it's definitely going to be there, and it will persist down south. So when we told New Zealand that it's coming, they're saying no, it's not going to be an issue. It's going to be die off during the winter, and 1 year later, it didn't die off, and they are talk that it couldn't possibly have been such huge population in such a small time. So it must be that we missed it when it first arrived. So the current still widely accepted, axiom is that like it's a pest that has long distance flight ability. So people have reported that if from the coast to somewhere central in the Caribbean with oil rigs over 100 miles, so 160 kilometers, they were able to trap it. So long distance flight ability, there's no arguing against about that. But how it actually so rapidly moving from West Africa all the way down to here in 3 to 5 years is quite amazing when you think about it. Does a flight ability really explain for that, given that you have Litura, you have up here you have Littoralis. These are all very related species. You have Armigera populations that are quite different from Australia. You don't get them. They're the same noctuid morph with the same flight ability. So why is it that this one all of a sudden spread so quickly and other native species have so far over eons remained pretty much in their native range? Something don't quite add up, unfortunately, and yet this is the general acceptance that this is a pest that is flying all the way from Africa, from pretty much a single introduction, because we look at a little TPI gene that tells us that they are identical. We look at a little bit of the CO1 gene, they are identical, so it's obviously a single introduction of very limited females, and they just spread very quickly. The folly there is that the markers that you're using are perhaps not the best marker to use. So that's why we go towards whole genome sequencing. So I currently also sit on the EFSA, so European Food and Safety Authority panel, because they're really worried about the spread of fall armyworms. So fall armyworms has been detected in, let me see, about there, Egypt, along the Niles. So water is really important and it's flying along the Niles. And that was 2017, 2018. And since then, it hasn't really moved up. So what's the situation? It's such a great flyer. Why isn't it there? But only very recently, when they approached me and say, help us to look at our management options and all that, and our going forward strategies, they say that it's actually in Cyprus now. So it's Southern Europe. So it's making its presence in Europe. And where would it actually end up in Europe? So a few studies look at that and say that these are possible worst scenario. But if we look at the climax, it's very similar. And this is not the worst scenario. This is just over years. And depending on what year it is and your rainfall pattern, it will have persistent populations in Europe. Like it or not, it's going to be there. And down south here and in Greece, there are big tomato crop. They grow tomatoes there, so it will be impacted eventually, I think. So global effort in focusing the spread of fall armyworms. So there have been quite a few. I'm just giving two examples. So this was Regan Early and Roger Day, so they're from CABI. And so CABI as the leading institution, and also FAO with help from CABI and US are saying that they are spreading from Africa and it's likely to be in Southeast Asia and so forth. So there is a lot of this talk about it's coming, prepare yourself. But where are the genomic evidence? You can say that they're coming. We do the way we anticipated. It's going to be coming, and you can see from news. They are spreading. Every time someone look at it, they found it. This is spreading and spreading and spreading. But really, if you look at the global map in Western Africa, you can't look to your west because it's Atlantic Ocean. So you can only look to your east, and every time you look towards your east, you find it. So is that spreading really fast, or is it because it's already there? And every time you look at it, there's not a country that look at it, didn't find it. So one of the examples is they look at the different studies, look at how wind pattern might help the spread of the fall armyworms. And in the Reagan study, they look at coming out from US and South America through passengers and trade routes. And they see that it's coming this way. But in that paper, they actually acknowledge that the spread pattern by natural means, using through your wind conditions and whatnot, is not actually very fitting because it doesn't fit the wind pattern of the moth flying towards the east. By 2018 May, India reported it, and then it went into Myanmar, Thailand by December 2018, and then it went into China, which is really important. They're producing 30% of the world maize. So it's very important. So it then spread all the way down south. Why is it important to get this right? So if we assume that this is all natural spread, you can't stop an insect mass when they're naturally spreading. There is no way you can stop it. You must just sit back, save your money. However, if this assumption is wrong and it's actually human-assisted, this is a behavioral trait, we can actually change that. We can minimize the risk. And after fall armyworm, there will be a lot of other pests coming. And if we just accept that it's always a natural spread pattern, then there's no hope because we can't stop and we haven't learned the lesson. So CSIRO, as I mentioned before, CSIRO very kindly gave some money to help me to start establishing this program to reassess the spread of the armyworms using whole genomics. So we wanted to know, and the underlying question is, what are the genomic signatures that we can use and we can identify to support the single introduction founder events in Africa that spread across the world? So do we have any other studies in the literature that actually identify single founders at the genomic level? Well, yes, we do. Tuta absoluta, the tomato leaf worms. It's a species from South America again, and first reported in Europe, and then spreading into Africa, and then gradually now reported in India and into China. So this is a fairly damaging pest, and we don't want it. And it's been spreading, and genetic studies suggest that they are actually single founder event. So they have very limited DNA pattern, if you like, signature. So every population is identical based on nuclear genes. So they are identical, suggesting that they are likely to be from a single stock, and then they all carry pretty much similar genetic signature, and it's spread around the world, and it doesn't matter where you sample the populations, you're going to see the same pattern. And that's what the single founder population genomic signature should look like. Admittedly, this is only a 5 DNA marker, but it's a nuclear marker, and this is what they found, and it does still fit more or less the general story of how this pest is spreading. So it's a big pest. I understand that there's a lot of tomato growers out here in our region. So we don't want that. We are going to be monitoring for that. We're trying to develop projects to find fundings to do that. What about the fall armyworms? When we look at the fall armyworms, if we were just looking at the full mitochondrial DNA, so the full maternal transmitted genome organelle, there's actually a lot of diversity in the native range, as you would see. So the white in this case represents just a unique. So it can be 3 unique that are different from this one, for example. Right? So there's quite a lot of unique one. But when you look at just some African populations from Benin and from East Africa and then India and China, you see that they don't fit the pattern of a single female founder because you have quite a lot of female founders in there. So this will suggest to us that the current studies that people have been using just a tiny bit of DNA is faulty. It's a lot of assumptions to make to then try to use that knowledge to develop management strategies. It's quite erroneous. And if we were to look at the genome signature, and you can see how different that is, these are different populations from countries, so from US to Central America to South America, and to East Africa, West Africa, East Africa, India, China, So that was our first study, and involved collaborators from China, from South America, from UK, from France, and a lot of CSIRO colleagues. And it showed that, in effect, the genomic diversity of the invasive pests across the invasive range from about here onwards are really, really diverse. It's not a single founder. These are multiple founders in different places. If you look really carefully, Chinese population, for example, is really, really quite different from African population. So in that sense, I would argue that China population didn't come from Africa. Despite the fact that you reported at the time that supposed to be coming from Myanmar, Myanmar supposed to be getting from India, and so forth, it did not match. Genomic signature tells us that they are different populations. So what's actually going on? These red dots are individuals that show that have very little or no genetic admixture, so there's no mixing. So if you have African populations that are hybrids, it doesn't matter how you breed across the populations, across the generations. You can't come back to a full breed, a purebred. So again, the genomic profile do not fit. And if we actually just look at how the markers, so we have 870 nuclear, so the nuclear genes and the neutral markers, and we can map the population. So this is a bit challenging to look at it, but essentially Brazil had two populations. This population is actually resistant to cryotoxin. Then you have another population that's here, and then you have US population in, let me see, it was Florida and Mississippi, and then Mexico. So we can say that this actually kind of match what we expect at the geographic distribution of the pest. They're quite different. And when we look at populations from Peru, Peru is right next to Brazil, it actually match here, sitting together with Brazil. So it does make sense. And then if we look at, and some Peruvian individuals are up there as well, I think. So it's a bit, there's some movements. And then if we look at Malawi, Malawi being East Africa, is sitting similar. So we're not saying that it is from Brazil because there's so many different countries now, South America that we haven't sampled, but it's very similar to the Brazilian and Peruvian population. So if Malawi, which is on East Africa, is similar to this signature, Benin being on the west is the original population, you would expect Benin to be right here. And when we did that, Benin sits here. Benin is quite different from West Africa. So we're looking at many different pathways for this pest to be spreading around the world. And that the idea that it came in from a single population with the same genetic signature across the world, and if they have a management strategy in West Africa, this is what we can expect. That's a folly. That's a mistake. You shouldn't expect that. So at the end of the day, when we do a gene flow analysis, so the directionality of how the gene flow is, the genetic markers are sharing across the population, it shows us that China, in this case, not China per se, Asia. But we only put China because those are the populations from China. So Asia as a whole is a fairly weak biosecurity hotspot because they were the one that actually went into Africa. So it's the other way around. Okay? And we have some movement in Peru and southern US as expected, but multiple Chinese populations were seen to have moved into Africa. So that's where I'm going to end. And later I'll talk about how we analyze the spread pathway of this pest coming from Southeast Asia into Australia and into New Zealand, perhaps, and what we can do going forward, I guess. But B will look at, we'll talk to you about the management options.